Hi everybody, this is my summary of project-based lesson. Sorry, that's our bell ringing. Okay, so for my project-based lesson, it was around Unit D, which is our cellular design in DNA. The objective that it mainly focused on was exploring the structure of DNA and its relationship to the cell. Prior knowledge for this objective was analyze cellular design and function in plant, animal, and bacteria cells. So first we talk about cells and their structure and the different organelles inside the cells and the jobs that they have. And then we narrow it down to talking about the nucleus, which is where DNA is located inside the cell. So once we get to the end of our cell unit, we move into DNA and start talking about the structure of DNA. Like I said, we start off with the history of DNA, how they discovered it, how they knew that DNA was our genetic material. We talk about DNA structure and that it's made up of nucleotides. Then we talk about the replication of DNA. Replication of DNA, we talk about how the DNA splits and starts to create two new strands of DNA. Then we go into talking about DNA damage and repair, focusing on mutation and how DNA has the ability to self-check itself and trying to repair some of those mutations before they can cause any physical damage to our well-being. So once we kind of do lecture and go into the fundamentals of DNA, we get to start having some fun, and we'll do an extraction of DNA lab, which is where we extract the DNA out of a strawberry.
Oh.
And the strawberry, the way that we break it up is we put it inside a Ziploc bag, and there's a buffer that we break the solution with, and that breaks the cell walls of the cells that's in the strawberry to release the DNA so that students can actually see the DNA of the strawberry. Then we'll create 3D DNA models inside the classroom as a class, and then they'll be assigned to create a 3D DNA model at home to make sure that they fully have understood the structure of DNA, how it's composed, and what it's made of. And then finally we have just a brief, assessment. I wouldn't call it a test because it doesn't have that many questions on it. I usually do quizzes at the end of the objectives to make sure that students really understand the objective and the topic that we discussed. So as I stated, students use their prior knowledge of cell structure to relate to where DNA is found in the cell. DNA is the genetic material of our cells. In students, the new knowledge that they gain that they haven't learned yet is the DNA structure, the replication, and the mutation. This is completely new to them. They haven't even discussed it in their science classes yet. The good thing about the cell unit is that it's right in hand with their cell unit that they're doing inside their science classes as well. So the students always enjoy that they're learning about it in their science class, and then they come into my class and they get to kind of get a second dose of learning about it, and students say that they enjoy how they know more and they feel more confident about the topic. And then with the knowledge that they gain, that's when they start to create their models and show that they really understand the structure of DNA. Here are some pictures of the DNA models that we make in the classroom. We just use different color toothpicks, and the different color toothpicks represent different structures of the DNA model. So the blue-green color toothpick here represents the sugar phosphate backbone of the DNA, and then the regular colored toothpick here represents the hydrogen bonds that are in between our base pairs, which are represented by dots candy. They can choose the colors that they want to represent of the 4 base pairs. So, A bonds to T and C bonds to G. And they create a little table for me and tell me what each part of their model is and what it does. And this part is done in the classroom before we move on to the extraction lab to make sure that they really understand the structure of the DNA. These are some of the at-home models that have been turned into me. This one is actually from last year's students, but it's one of my favorite models. I still have it on my desk to this day. This is another one that a student created and turned in a little table to show what each area is, and here's another creative way. And so I really encourage the students to be creative, use what they have at home if they can, so that they don't have to necessarily go to the store to create the model, and they can be more creative and figure out what they have at home that they could use to create it. Then in our DNA extraction lab, which the students really enjoy, we start off, like I said, we put a strawberry inside a Ziploc bag, and there's a buffer solution that we put inside that bag, and the students then have to smush the strawberry, so they squish it in their hands to break the cell wall, let the buffer solution get into the strawberry and break it all apart. And we'll pierce a hole in the corner of the Ziploc bag to funnel and strain out all the clumps, which gives you this end product here that you see in the middle. And so as you can see, it shows the separation, and what happens with the buffer is that the DNA will start rising to the top of the test tube, and the students have the ability to use a micropipette to pull that DNA out and put it into this little miniature centrifuge tube, and they get to leave the classroom wearing that around the neck with their strawberry DNA inside of it for them to kind of show their other teachers and their parents what they did in my class that day. This unit is very exciting and fun for me. I really enjoy this unit. This in the cell unit is one of my favorite units to teach my students. They get really engaged and they enjoy it, and they're fascinated by it, and they ask a lot of questions, which I absolutely love. And that is my project-based lesson. Like I said, after the extraction lab, we'll end with just a quiz, kind of going over all the main topics that we discussed to make sure that the students have got a full grasp on the DNA structure and the replication and mutation. Thank you so much. I hope you all have a great evening, and thank you for the feedback.